Hello, welcome to this Dr. Rajiv's lecture on gene editing. This course on gene editing is designed for UG and PG students of medical genetics, agriculture genetics, biogenetics, biomedical engineering, genetic engineering, biotechnology, as well as for research scholars and young scientists. The genome of an organism has always fascinated researchers. The discovery of restriction endonucleases enabled scientists to make targeted manipulations or knockouts in any gene sequence of any organism by the technique popularly known as genome engineering. But today is an era of gene editing dominated by the CRISPR-Cas9 tool due to its ease of design and handling. This course on gene editing introduces you to the basic features and applications of gene editing, base editing, and CRISPR technology. This lecture covers topics like zinc finger nucleases, TALENs, CRISPR system, and the latest techniques like base editing and prime editing. Issues covered in this course are: what is gene or genome editing? The difference between gene editing and genetic engineering. Usage of gene editing today. What is the chronology of gene editing tools and techniques? Discussion on zinc finger nucleases. Discussion on transcription activator like effector nucleases. Discussion on CRISPR-Cas9 system. Discussion on base editing. Discussion on prime editing. Comparison of basic genome editing tools. The scope of human genome editing. The role of artificial intelligence in gene editing. Scenario of gene editing in India. And ethical and safety concerns about gene editing. The gene editing or genome editing with programmable nucleases is a rapidly growing field in genome engineering. It involves transformative technologies that allow researchers to make precise changes to the DNA of a living organism for numerous applications such as biomedical research, biotechnology, agriculture, human gene therapy, and patient treatment. The gene editing is a sophisticated type of genetic engineering that can add, remove, or alter genetic material at specific locations within an organism's genome. In gene editing, there are two basic processes: the cutting technique, performed artificially, and the repair mechanism initiated by the cutting and realized naturally. Although many gene editing techniques exist, all are based on the use of enzymes called nucleases. Nucleases can be redirected to target a desired site on DNA within the genome. After the site-directed nuclease recognizes the locus of interest, it binds to the target site and generates a DNA strand through several editing processes. Gene editing modifies an organism's genetic code through a set of tools and specialized techniques. The most notable and widely used gene editing technology is CRISPR-Cas9 system. It allows for precise modifications to the DNA of living organisms. The four basic tasks performed in gene editing through programmable nucleases are recognition of target genomic loci, binding of effector DNA binding domain, double strand breaks in target DNA by the restriction endonucleases like FOC1 and Cas, and repair of double strand breaks through homology directed recombination or non-homologous end joining. Genome editing techniques like CRISPR, TALENs, and ZFNs are now in widespread use to alter specific sequences in genomes of cultured cells. What is the difference between gene editing and genetic engineering? Gene editing and genetic engineering both are used to make changes in the genetic material of organisms, but they differ in their methods and precision. The current genetic engineering technologies precisely modify the DNA to mimic the naturally occurring differences in DNA sequence. They randomly insert genetic material into a host genome, whereas gene editing targets site-specific locations. The genetic engineering involves the direct manipulation of an organism's DNA using various techniques such as inserting genes from one organism into another to confer new traits like pest resistance in crops, modifying existing genes to enhance or suppress certain characteristics, creating genetically modified organisms or GMOs, which can involve adding, deleting, or altering genes in a way that may not be precise. The gene editing, on the other hand, is a more precise form of genetic engineering. It targets changes to the DNA sequence at specific locations. The CRISPR-Cas9 technique of gene editing allows deletion, replacement, or adding DNA at precise loci in the genome. It edits specific genes without introducing foreign DNA, which is a more refined approach. Key differences lie in precision, techniques, and applications. The gene editing is more precise at targeting specific DNA sequences, while genetic engineering is broader and less specific. Gene editing uses precision tools like CRISPR-Cas9 for targeted modifications, whereas genetic engineering may use older and less precise techniques. Both are used in agriculture, medicine, and research, but gene editing is often preferred due to its accuracy and reduced risk of unwanted effects. The gene editing is a powerful tool that has a wide range of applications across various fields. Some of the key areas of application are medicine, agriculture, environmental protection, biofuels, and basic research. Scientists use gene editing to develop treatments for genetic disorders such as muscular dystrophy, cystic fibrosis, and sickle cell anemia. CRISPR based therapies have shown promising results in clinical trials to treat these conditions. In agriculture, the gene editing helps create disease, pest, and environmental stress resistant plants. It also enhances yields and the nutritional value of crops. Gene editing techniques can introduce precisely targeted modifications in any crop. The gene editing is used to manage harmful species and control disease vectors like mosquitoes that spread malaria and dengue. This can help protect ecosystems and reduce the transmission of diseases in humans and crops both. Gene edited microbes break down agricultural and industrial waste into biofuels. This not only provides a renewable energy source but also helps in waste management and environmental protection. Scientists are using gene editing to modify microorganisms like algae and bacteria to produce biofuels at a higher rate and with greater yield. The gene editing technology can reduce the need for chemical inputs to produce biofuels and make the production process more sustainable and environment friendly. Scientists use gene editing to understand the fundamental aspects of biology and genetics, including gene function studies, disease models, genetic pathways, drug development, and evolutionary biology. This can lead to new discoveries and innovations in biology, agriculture, and medicine, such as inserting a new gene to make organisms produce useful medicine, treating genetic diseases, creating tailor-made organisms to study human diseases, and producing replacements for damaged or diseased tissues and organs. The evolution of genome engineering started about 7 decades ago with the discovery of the DNA double helix by James Watson and Francis Crick. In fact, research in this field has been active since the 1970s. During these years, researchers uncovered different techniques to perform gene editing. The journey of the gene editing starts with meganucleases and zinc finger nucleases, walked through transcription activator like effector nucleases or TALENs and CRISPR-Cas9 to reach base editing and prime editing. Meganucleases, discovered in 1990s, were among the first tools used for gene editing. They are natural enzymes that recognize and cut specific DNA sequences. But their use was limited due to the difficulty in targeting new sequences. Precise gene editing technique began with the introduction of zinc finger nucleases or ZFNs again in 1990s. Zinc finger nucleases are artificially produced restriction enzymes for custom site-specific gene editing. TALENs or transcription activator-like effector nucleases came in 2011 are a molecular tool derived from a plant pathogen. These are capable of single nucleotide recognition, thereby increasing targeting capabilities and specificity compared to zinc finger nucleases. Talents are cheaper, safer, more efficient, and capable of targeting a specified region in the genome as well. In 1987, an important tool, clustered regularly interspaced short palindromic repeats or CRISPR, was discovered in Escherichia coli by Isino and colleagues. However, the development of the CRISPR-Cas9 gene editing technology is credited to Jennifer Doudna and Emmanuel Carpentier. They published their groundbreaking work in 2012 and won the Nobel Prize of Chemistry in 2020 for this work. The CRISPR technology is based on a guide RNA and the CRISPR-associated protein 9 or Cas9 endonuclease, hence called CRISPR-Cas9 system. The base editing technology developed in 2016. Unlike previous techniques, it avoids cleavage of nucleic acid backbones and directly modifies the target nucleobases. It offers a more precise way to correct point mutations. The latest in the armamentarium is the prime editing that came in the year 2019. Compared to base editing, the prime editing is a more versatile molecular tool for gene editing. It can precisely perform targeted insertions, deletions, and base exchanges while limiting unwanted effects without causing double-strand breaks. Zinc finger nucleases are engineered DNA binding proteins. Zinc fingers were discovered in 1985. Efficient gene editing relies on a site-specific DNA double-strand break. Zinc finger nucleases were developed to generate a site-specific double-strand break at a desired genomic site. Each zinc finger is a domain stabilized by a zinc ion and a hydrophobic core, allowing for specific DNA recognition. They facilitate targeted editing of genome by creating double-strand breaks in DNA at user-specified locations, which is then repaired by the cell's natural repair mechanism, leading to targeted changes in the genome. ZFNs are composed of two main parts, a zinc finger DNA binding domain or ZF protein and a FOC1 nuclease domain. Each zinc finger protein or ZF protein can recognize a sequence of 3 to 4 base pairs, and multiple zinc fingers can be combined to target longer sequences. The first part recognizes and binds to a specific DNA sequences, and the second part cuts the DNA. It requires dimerization or pairing with another FOC1 domain to become active, ensuring that DNA is only cut when two ZFNs bind to adjacent sites on the DNA. When two ZFNs bind to their target DNA sequences, the FOC1 domains come together and create a double strand break. The cell then repairs this break using one of the two methods. One is non-homologous end joining (NHEJ) and the other is homology directed repair (HDR). The NHEJ can introduce small deletions or insertions, potentially disrupting the target gene. The HDR uses a repair template to make precise changes to the DNA sequence. Among applications of ZFNs are gene therapy, modifying crops, and gene study. Zinc finger nucleases are one of the earlier tools developed for genome editing, alongside other technologies like TALENs and CRISPR-Cas9. TALENs are transcription activator-like effector nucleases. These are precise and efficient gene editing tools. TALENs consist of a DNA binding domain linked to a specific DNA cleavage domain, usually a nuclease like FOC1, to cut specific DNA sequences. TALEN gene editing technology is useful when no suitable CRISPR-Cas sites are available for a specific design. It may also be more efficient than CRISPR-Cas9 in editing hard-to-edit genome regions such as heterochromatin. Talents exhibit superior homology-directed repair efficiency due to closer proximity to the insertion site. The key advantages of talents are customizable binding and no palm site requirement. Hence, tall effectors can be engineered to bind almost any DNA sequence, making talents highly versatile and allowing for more flexible targeting. Talents are used in plants, animals, and human cells for tasks like gene knockout, gene insertion, and gene editing. The CRISPR gene editing has evolved as one of the humanity's most powerful technologies. The CRISPR-Cas9 system is the most flexible and user-friendly tool for genome editing. It eliminates the need for making new proteins to recognize each new target site. It is less expensive and relatively easy to design and use compared to older technologies such as zinc finger nucleases. This is one reason why CRISPR is a game-changing technology. In the first decade of 21st century, the scientific community understood its ability to recognize specific genome sequences and cut them with CRISPR-associated protein 9 or Cas9 protein. The Cas9 works with CRISPR and has DNA cutting abilities. It inserts a small pieces of edited DNA at areas of choice along another DNA strand. In this way, CRISPR-Cas9 is a customizable tool in gene editing. The CRISPR is naturally used by bacteria as their immune system to kill invading viruses, and it has now been adapted for use in the gene lab. The CRISPR system has two basic components, a Cas9 protein and a guide RNA or gRNA. The Cas9 protein acts like a wrench, and the specific RNA guides, known as CRISPR, act as a set of different socket heads. The guide RNA directs the Cas9 protein to the correct gene or area on the DNA strand that controls a particular trait. This allows researchers to study our genes in a specific and targeted way in real time. The guide RNA is a small strand of RNA programmed to look for a specific DNA sequence. The Cas9 enzyme is a strong cutting device that can cut through a double strand of DNA. The guide RNA and Cas9 remain connected. When put inside a cell, the CRISPR system locates the target DNA. A CRISPR seeking device recognizes and binds to this target DNA. Then the enzyme Cas9 acts quickly and cuts the double-stranded DNA at two places, removing the desired specific piece to create an edited DNA. Scientists can now insert these new edited sections of DNA into the cell. The cell incorporates the new DNA into the gap automatically when it repairs the broken DNA. The CRISPR can also cause deletions in the genome and can insert new DNA sequences. Many researchers have observed that the CRISPR is 6 times more effective than ZFNs or TALENs in creating targeted mutations in the genome. As a result, the large genomic projects that once took many years and huge funds can now be completed in a small fraction of time and cost. Base editing is an advanced type of gene editing technique. It allows for precise changes to individual DNA bases without cutting the DNA strands. The base editing method is particularly useful for correcting single point mutations, which are responsible for many genetic diseases. Base editing uses a modified version of the CRISPR-Cas9 system known as Cas9 nickase. Instead of creating double strand breaks, the Cas9 nickase only nicks one strand of the DNA. The guide RNA directs the Cas9 nickase to the specific DNA sequence where the deaminase enzyme makes the desired base change. The base editing offers several advantages compared to other gene editing techniques like traditional CRISPR-Cas9. Base editing allows for precise single base changes without creating double strand breaks in the DNA. This reduces the risk of unintended insertions or deletions and chromosomal damage. As stated above, it uses a modified CRISPR-Cas9 system combined with a deaminase enzyme to convert one DNA base into another, for example, cytosine to thymine or adenine to guanine. One notable example of a successful application of base editing is the treatment of progeria. The prime editing is a state-of-the-art gene editing technology. It is often described as a search and replace tool for the genome. Prime editing combines the precision of base editing with the versatility of traditional CRISPR-Cas9 technique. It can make targeted insertions, deletions, and all 12 possible base-to-base conversions without creating double-strand breaks in the DNA. Prime editing uses a modified CRISPR-Cas9 nickase and a reverse transcriptase enzyme to directly write new genetic information into the DNA. It is suitable for a wide range of genetic corrections, including those that require more complex edits than base editing can achieve. The human genome editing is editing of nuclear DNA in a way that may be heritable across generations. Heritable human genome editing has been a subject of intense debate due to its possible consequences for offspring and for society. These technologies can be used on somatic cells as well as on germline cells. The human genome editing, particularly using CRISPR-Cas9 technology, has numerous possibilities in medical research and treatment. Some notable examples and applications include treatment of genetic disorders, HIV treatment, and cancer research. Researchers have used CRISPR to correct the mutation that causes sickle cell disease in bone marrow stem cells. These edited cells are then reintroduced into the patient's bone marrow to induce the production of healthy red blood cells. Efforts are underway to use CRISPR to correct genetic mutations responsible for cystic fibrosis, potentially offering a cure for this lethal condition. Gene editing is used to create more accurate cancer models, helping researchers understand the disease better and develop targeted therapies. CRISPR technique is being explored to remove HIV DNA from infected cells or to modify the genome of immune cells to make them resistant to HIV infection. CRISPR has been used in animal models to correct mutations causing Duchenne muscular dystrophy, showing promise for its treatment. To treat inherited retinal diseases such as liver congenital amaurosis, which causes blindness, is also the focus of CRISPR research. Research is on to edit germline cells and stem cells like sperm, eggs, and embryos to prevent hereditary diseases. This approach could potentially eliminate genetic disorders from future generations. The artificial intelligence is helping us with everything from identifying abnormal heart rhythms before they appear to detecting skin cancers. AI gene editing refers to the integration of artificial intelligence with gene editing technologies like CRISPR and zinc finger nucleases to enhance precision, efficiency, and outcomes in genetic modifications. AI algorithms can analyze vast genomic datasets to identify optimal target sites for gene editing, minimizing negative effects and ensuring more accurate modifications. AI can design more effective guide RNAs and proteins for CRISPR systems, such as the AI designed protein OpenCRISPR-1. Now it is becoming possible that the artificial intelligence may enable the production of customizable proteins called zinc fingers to treat diseases by turning genes on and off. The researchers at NYU Grossman School of Medicine of New York University and the University of Toronto designed a tool that promises to accelerate the development of gene therapies on a large scale. The convergence of AI and conventional gene editing holds great promise for advancing genetic research and shaping new therapies. The gene editing is gaining momentum in India across various fields, including healthcare and agriculture. The sickle cell gene editing mission in India is focusing on developing gene editing therapies for genetic diseases like sickle cell anemia and beta thalassemia. This mission is supported by various government agencies, including the Council for Scientific and Industrial Research (CSIR) and the Indian Council of Medical Research (ICMR). Institutions like the Institute of Genomics and Integrative Biology in New Delhi and the Narayana Nethralaya Foundation in Bengaluru are actively involved in preclinical and clinical trials for gene editing therapies. India has recently relaxed regulations on gene edited crops, allowing field trials of CRISPR-Cas9 modified rice variants. This aims to improve crop traits without adding new genetic material. There are as many as 16 institutions in India engaged in gene editing research for agriculture, focusing on developing crops with improved disease resistance, climate resilience, and nutritional quality. Indian scientists at the CSIR Institute of Genomics and Integrative Biology in New Delhi, along with the LV Prasad Eye Institute, made a significant breakthrough in gene editing. They developed an enhanced version of the CRISPR-Cas9 system known as FN-Cas9. They claim that FN-Cas9 tool is more precise and efficient than existing CRISPR system. While somatic cell editing is progressing, germline gene editing remains banned in India. There is ongoing debate about the ethical and legal implications of these technologies. There are numerous scientific, safety, ethical, and policy concerns today in researchers' mind and governments. They tell that the genome editing must be safe before it is used to treat patients. Serious safety and ethical issues are there in altering the germ line because edits in the germ line would be passed down through generations. Also, a current lack of compelling medical applications do not justify the use of genome editing in embryos. Many countries and organizations have strict regulations to prevent germline editing for these reasons. In 2017, the National Academies of Science, Engineering and Medicine (NASEM) recommended that clinical trials using gene editing in embryos should be permitted only within a robust and effective regulatory framework and only when certain criteria are met. The International Commission concluded in the report Heritable Human Genome Editing, released in September 2020, that clinical use of heritable human gene editing should not be considered until it has been established that it is possible to make precise genomic changes efficiently and reliably without undesired changes in human embryos. The Dickey-Wicker Amendment prohibits the use of congressionally appropriated funds for the creation of human embryos for research purposes or for research in which human embryos are destroyed. If permitted, use should be limited to serious monogenic diseases. The potential effects of gene drives on individual species and the environment have also raised biosafety, biosecurity, and ethical concerns.